I went overseas in 1961 and taught in England for 2 years, then got married and moved to Seville. And while in Seville, I applied for a National Science Foundation summer study grant, and it was in ecology. It was for biology teachers. And I went. So this is, I believe, in 1964, that summer, where I went to the University of Rhode Island at Kingston. And there were a large group of biology teachers from all over the United States. And one of the things we did was to go on the ship Trident. That was the oceanographic vessel that the University of Rhode Island had. And then later we took many field trips along with classroom lectures and studies. Then we did our own studies too. So this is a little picture show of it. There's Rhode Island in the background. We're in Long Island Sound. And this is the boat. Ship, I guess. It was a big boat. And there are the teachers. We brought our lunches and we knew what was going to happen. But one of the first things we did was to get an idea of what goes on board a ship like this. And of course, it's to study coastal waters and the uh, ecology of the sea. This was the first thing we looked at, which is a device that's dropped overboard. It's run in this horizontal position like this. So as the ship moves along, water enters this end, and on the other end there's a net that lets the water out, but then a collecting device to capture any microorganisms that don't fit through the net, and the net is fine mesh. So this, in essence, is getting plankton, which is the microscopic organisms of the sea, which is the first food item of many that feed on this and then build up through the food chain. So here it is being placed overboard, and it's run for a while. So there's the capturing device, and that fits at the end. And then at the end of this, there's a little collection device. This was the zoologist in charge of this experiment, and that's what they got. So again, you can figure out the speed of the boat, the amount of water that went through, and what was collected. And this gives you then a sampling of what is expected in this kind of water at that depth in Long Island Sound. Okay, we did go down later to look at some of these under the microscope, and they were the typical kind of plankton organisms, protozoans and diatoms and that type of thing. And you get to see why these booms that are on the boat are used to do various things. Now you might wonder, what was the boat doing out there that day? Was it just taking this group of teachers for a field trip? No, it actually had a function. It had a device that trailed behind the boat, sending out electronic signals, and they were looking to sort of map the bottom of the ocean there, or the sound, and to see where rivers might have gone, and checking out the topography in a sense. And that was the device that they used for this. Now, another device that they use, and of course I'm assuming today many of these are much more advanced, but this is what we had back in 1964. This was an oxygen sensing device that could tell the percent of oxygen in the water. Again, I don't know the electronics, but that was then telemetered and was recorded. Now you might say, why is oxygen important? Well, things that live in the ocean need oxygen to respire. There are some anaerobic organisms that don't need it, but most of them are aerobic, and the amount of oxygen is critical. Now, this device right here is something that's dropped from the boat, and then it is driven into the mud. So it then gives you a core of sampling from the bottom where the boat happens to be. And then you could study this and look at it for either fossil evidence or microscopic evidence of organisms or whatever they do with this type of data. This device is also interesting. A series of these were set along this cable with a weight at the bottom, and it was dropped down. And then they run a slider down this wire, and it opens up and it captures the water at 10 feet, 20 feet, 30 feet, 40 feet. And then this water is then taken and they can be specifically analyzed. Again, these are tools of the ecologists and what they do. Now, this one's very primitive and does exactly what you think it might do. It's dropped down to the bottom of the uh, ocean there, and we weren't in very, very deep water, and it just scoops up a section of what's there. It's brought up to the surface, and then that's analyzed. You don't have to send anyone down there. You can bring that stuff up and analyze it. And again, all of this is collecting data to give you an idea of what is there. And then the material is put in this little bucket, and depending upon what they're looking for or what they're going to record, becomes, as I said, data. Now, a little diversion we had is that while we were out there, there were trials for a race, a yachting race, and we saw some of that. Okay. Again, it was a nice, relatively calm day. I don't think anyone got seasick, but a good time was had by all, and we learned something. And again, the tools of the trade. Now, I decided to put these two together so we could have one long oceanographic coastal ecology slideshow, so to speak. And here we're looking at coastal ecology and tidal pool ecology. And this was Professor Palmetier, very, very nice fella. And we would take our buckets and our little jars and our uh, nets and things to gather, and there we are off to collect. And there I was. I brought my camera, of course. All of these are my slides. And what we see here is an interesting uh, stratification of this coastal outcropping of rock that as the tide comes in, you see there's actually like horizontal lines. And we see in this area there is fucus, which is a seaweed, and that forms a band right about there. Now, just below this level, always just below the fucus level, is the mussel shells and mussels, a sort of clam. And then above that, you get what is called the blue-green algae band. So, twice a day, as a result of the tides, this is what happens. And of course, you do get waves that splash up. But these bands indicate specific areas along coastal areas like this where it shows what we call stratification due to particular uh, events of the tide going in and out. Now, depending on the slant of it, it'll be more concentrated if it's straight, but here it's sort of spread out a lot more. And then you can see what we see here, and we're looking at that. Here's rather steep, and again, you can just see the mussels just below the low tide zone. And I like to eat mussels, so. And then this is the fucus, the algae, and then this is the blue-green algae up in here. Now, another thing that's very common in some areas is barnacles. So we see lots of barnacles all along this particular area. And here's one that's looks like it was taken at a different time, but shows a kind of a stratification. But again, since it's not steep and this is much less slope, the areas are spread out. There is sort of a barnacle layer here as well. Okay, here's lots of little periwinkles, little snail-like organisms that were all over the place here. And there's a close-up of the barnacle layer here and the fucus. And then down below you would expect to find mussels. Now, tidal pools. The tide comes in and out, and depending on the nature of the rocky areas, there may be little basins that are left after the tide goes out. So these areas contain seawater, while the surrounding rock that's higher don't lose it as the tide goes out. So within these areas, these are called tidal pools. And what we did is, I'm not a specialist in this, but we looked at this and just look at the abundance of different types of organisms that are found in this tidal pool. Now you can figure out its height. It's probably only 6 inches of water here. While if we look at other tidal pools, might be much deeper, and there's some fucus and some other seaweeds here, and I'm not sure whether these are barnacles or what. Here's a very, very deep one, and I mean not too deep, but again, the variety of life that are found in these tidal pools. Here's one where it only seems to contain this sort of greenish algae. Again, I'm not a specialist on this, but some of our biology colleagues did choose this to study the different tidal pools. Here's I'm not sure whether this is a coral or a sponge formation, but I just threw this one in, and this one as well. I'm not sure what this is. In the old days, I may have remembered, but it's been a long time since I took this field trip. Now, here again, a little further out away from the rocks, we see actually kelp, which is a seaweed that forms long leaf-like strands. And Rachel Carson, who wrote the book Edge of the Sea, describes this as one of the most tremendous environments to live in terms of changing, where it's dry, it's wet, there's waves, different temperatures, and organisms survive there. And kelps, as you see, if they were rigid, they would be broken off through the wave actions, but they move and float. Off the coast of Florida, there's tremendous kelp beds, and that's what we see here. And then, interestingly, up on the rocks near the coastal area, we sometimes see little plants. This is not quite in focus, but It's just growing in that little crevice there, a little bit of soil, a seed, and it must be capable of handling salty water because that's probably what it gets a lot. And the pencil is used to show relative size here. Ah, uh, we were on a beach and people who were visiting the beach, especially children, came over to see what we were doing. And one of the things we found that is if we took a rock from a beach and then picked it up carefully, that under the rock you'd find many, many marvelous things. So here's some little starfish here. Now they're not really fish, they're echinoderms. There's a little periwinkle, there's some barnacles, and what a good ecologist would do then is carefully turn this upside down and put it back where you got it. We were just observing what was there. Now this young lady was out there interestingly working around and collecting things. We had our little bags that we collected things we could take it back to our lab to study. But shortly after I took this picture, she was bending down to pick something up from the water, and a wave came over and just flattened her out. Oh, that was a kind of an interesting thought. I said, "Here she is, nice and dry, but got to be careful around there. It's kind of an interesting environment." And there you can see the actions, and there perhaps are some small kelps or something on this rock. Again, The Edge of the Sea, Rachel Carson. I think you'll be so pleased to read it. Such a wonderful book, and she wrote so well. She's also the author of Silent Springs, which is given the title of the mother of modern day ecology, showing the dangers of the pesticide use in our environment. Now, if any teacher sees this, this was an interesting technique that they showed us that if you bring along some note cards and if you're knowledgeable and you sort of know what you are going to find, you might have the names pre-printed or written on these note cards. You write down characteristics of them and have the students find them and then make a little display of them afterwards. So these were all the kinds of things that people in the group knew. And again, this is not my area of expertise, but I thought a good teaching technique. Even if you're on a trail, in fact, I did use that, where we'd bring the cards and who can match up to species of trees or shrub or plant that we would find, and then, of course, with a camera, makes it a nice way to record the event. And that's what I looked like back in 1964. More hair, less weight. Here, this is a conch shell seed pods, I believe, that I was looking at, and I would carry my little uh, magnifying glass to look at things really well. So a typical scientist. And here's just a little summary of what I've said already. And as a result of this, by the way, this 6-week workshop in ecology, as I taught biology over the years, I've always featured the ecology part and did our summer programs. And so again, I think the money that the National Science Foundation spent on teachers training them for this was money well spent.